Right now, I am going to start another topic from genetics, that is the crossing over. Let me first explain what is crossing over. Crossing over means exchange of chromosomal segments between two non-sister chromatids of a pair of homologous chromosomes. So, mind it, the chromatids should be non-sister, but chromosomes should be homologous. And you people might have studied in cell division that always synapsis of chromosome or pairing of chromosome occurs at zygote substage of meiosis 1, whereas crossing over occurs at the Packetin substage of meiosis 1. So, in crossing over, the chromatids should be non-sister and chromosomes should be the homologous one. And always crossing over occurs at tetrad stage, and this tetrad stage is clearly visible at Packetin substage of cell division. So, suppose this is the maternal chromosome and this is the paternal chromosome. This is the maternal chromosome, this is the paternal chromosome, and it is the centromere. And in between these two, this synaptonymal complex appear. This is the synaptonymal complex. And synaptonymal complex forms the physical basis for the pairing of this homologous chromosome. Here one enzyme acts, and the enzyme is recombinase. This recombinase, or you can artificially call it restriction endonuclease. I am just writing this enzyme just to clear the points. Recombinase do the same thing which restriction endonuclease do in the case of biotechnology experiment or genetic engineering experiment. So, here suppose breakage occurs in the chromosome number. This is suppose chromatid number 1, 2, 3, 4. So, 1 and 2 is sister chromatids, 3 and 4 is sister chromatids. So, if chromosomal exchange takes place between 1 and 2 or 3 and 4, it is not called crossing over. So, for crossing over, the exchange of chromosomal segment must be between 2 and 3 or 2 and 4 or 1 and 3 or 1 and 4. So, suppose here breakage occurs in chromosome number or chromatid number 2 and 3. 2 and 3 the breakage occur. If breakage occurs in 2 and 3, and it is basically joined by the synaptonymal complex. This is maternal chromosome, this is paternal one, 1, 2, 3, 4. Now, during exchange they cross each other. They cross each other means they form a cross-like structure. They form a cross-like structure like this. Like this. This one exchange with this and this one exchange with this. And this in between the two, this synaptonymal complex is present. And this cross-like structure is called chiasmata. And from here crossing over occurs. And this crossing over resulted into genetic recombination. So, in genetics, wherever we call recombinant class, it always the result of the crossing over. And this mechanism is called breakage and reunion theory. This is called breakage and reunion theory proposed by Darlington. This is called breakage and reunion theory, and it was proposed by Darlington. This theory differs from other theories in fact that other theories suggest that chiasmata is the cause of crossing over, but this theory clearly indicated that or clearly proved that chiasmata is the result of crossing over, it is not the cause of crossing over. So, you can easily see here, first crossing over occurs, then chiasmata forms. It means this chiasmata is the result of crossing over, this is not the cause of crossing over, and this is all about the breakage and reunion theory of crossing over.